All right, so on the day we went over lesson 5, we talked about the importance of compartmentalization in eukaryotic organisms, right? We didn't spend a lot of time talking about what it means to be a eukaryote versus a prokaryote. Okay, so today we're going to talk a bit more about what that means, kind of what the importance of compartmentalization is in terms of the difference it makes between a prokaryote and a eukaryote, because we didn't talk about that part of it. Okay, we said compartmentalization is important because it creates these different environments within the cell, but we didn't look much at it beyond that. Okay, so we need to be able to identify and describe the characteristics of a prokaryote, same for a eukaryote, okay, and be able to distinguish between the two types of cells. It's fairly easy to distinguish between the two. Prokaryotes are generally much smaller than eukaryotic organisms. Okay, they also tend to be a lot simpler. And even if you can, like, blow them up to a size where you can see them well, there isn't much to see because they don't have the organelles, okay, that a eukaryotic organism would have. So it's quite easy to tell the difference between them. Okay, most prokaryotic organisms are bacteria. Okay, essentially all of them are bacteria. That's what a prokaryote would be. Okay, so we'll kind of look at what they mean. All right, so bacteria are prokaryotes, that is, they are single-celled. There are no multicellular prokaryotic organisms. Why not? What do you need to have in order to become a multicellular organism? Okay, compartmentalization, and your cells have to become specialized. All right, if you don't have any organelles within your cells, how do they become specialized? They can't. Okay, if you're looking at muscle cells, they need lots of cytoskeleton, mitochondria. Okay, they're actually multinucleate as well. Okay, a muscle cell may have several nuclei inside of it, so that when it tears, okay, it can go back together. It has a nucleus in both of the torn sections and such. Okay, so you can't become specialized if you don't have organelles inside. All right, so most, if not all, prokaryotes have cell walls. Okay, these cell walls are not, they are analogous to plant cell walls. That means they are similar in their purpose, but very different in their structure. Okay, and in fact, it's the cell walls of bacteria, okay, that we attack with antibiotics. Okay, most bacteria are susceptible to basic antibiotics like penicillin. Some are not. Okay, some are penicillin resistant. Right, that has to do with the structure of their cell wall. There's a simple mutation that can happen. Okay, but if their cell wall doesn't contain a certain compound, then penicillin can't dissolve their cell walls. Okay, and then obviously it doesn't kill them. Okay, so what you see here are essentially the three groups or shapes that bacteria can come in. Okay, they can come in spheres, rods, or spirals. Okay. All right, so there's diversity in the shapes. Okay, the three most common being spheres, which we call cocci, C-O-C-C-I. Okay, Streptococcus mutans, okay, causes strep throat. Okay, people have ever had strep throat. Okay, so that's Streptococcus mutans. Okay, there are other versions of that that can cause more serious infections. Okay, like the flesh-eating bacteria. Okay, is a mutated form of Streptococcus mutans. Okay, and that can lead to all sorts of problems. Rod-shaped, okay, so bacilli are what we call the rod-shaped ones, okay. The tetanus is a example of that, right? Salmonella is also a bacillus bacteria. So if you've eaten at Arby's, okay, you might have probably got that. Okay, I always bash Arby's. I've eaten there twice. You know how many times I got food poisoning there? Twice. I just assume that you always get it when you go there now because I figure that's a safe assumption. I'm 2 for 2. I haven't eaten there in 25 years, and I never intend to eat there again. Okay? Fool me once, shame on me. Fool me twice, no way. It's the other way around. But you know what? You get food poisoning once, never eat at that place again. Okay? I should never have eaten there again, but I did. Stupid. Okay, so yeah, they are a bacillus bacteria. Okay, they're susceptible to antibiotics as well. Okay, you can get rid of them that way. And then there are spirilla. Okay, that's the spiral bacteria. Treponema pallidum causes syphilis. Okay, in fact, a lot of the bacteria-typed sexually transmitted diseases are spirillae bacteria. Okay. All right, so those are the different shapes. Okay, without knowing a whole lot about them, you can't, like, if you were to look at them under a microscope, anything that's a cocci bacteria looks spherical. Okay, you really have to know your stuff to be able to tell one type of coccus bacteria from another. Okay, so bacteria can be sensitive to salt and temperature. Okay, so in order to kill bacteria, we usually use soaps or high temperatures or high pressures. Okay, things like that. Bleach obviously, okay, can kill them off. If we want to prevent them from growing on food, we need to make it difficult for them to grow. So we create conditions within the food that are hard on bacteria that could make us sick. Okay, that is, we could dry it out. Okay, so if you've cured, how many people have ever made jerky before? Okay, so when we make jerky, right, we add curing salt to it, right? That curing salt helps to dry it out, but it also makes it a little less palatable for bacteria, so it can stay, let's say, fresh for longer. Okay, it won't spoil, because bacteria can't get as good a foothold in it if it's A, dry, and B, really salty, or at least the kind that could hurt us can't get a foothold on it. All right, so what you see here on the right-hand side is a petri dish. Okay, and a petri dish is what you use to grow bacteria. All right, so if you've ever had a throat swab, okay, they stick the Q-tip down your throat and then they smear it on a petri dish. Okay, they put the petri dish in an incubator. The incubator keeps the petri dish at 37 degrees Celsius, so it's the same as your body temperature. And if you have any bacteria in there, they'll grow on the petri dish. And then what they do is they take a sample of them and they look at them under the microscope and they see what kind of bacteria you have, and then they tell you whether you need antibiotics or which antibiotics you need. Right? This is what it ends up looking like afterwards. So each one of these colonies, okay, these little dots, is the result of one bacterial cell being placed there by the Q-tip. Okay, just one. Over the course of only a couple of days, that single cell divides over and over and over again until there's a big enough colony of them or community of them that you can visibly see it. Okay, so now you know why you get sick so fast. Okay, they can reproduce pretty quickly. All right, they're tiny little microscopic things, yet they make something big enough for you to see. All right, this picture on the left, what do you suppose that is? Well, there's some bacteria there. There's also some fungus there. That's an electron microscope photograph of a cutting board. Okay. Yeah, ew is right. Okay. If all you do each time you use your cutting board is rinse it off, that's going to be your cutting board eventually. Okay. It's also why you should have a separate cutting board for meats, especially chicken. Okay. Then you don't want to cut your chicken on a cutting board and then go, oh, I'm making stir fry, grab the carrots and whatever else and start chopping them on the same cutting board. You're gonna get sick. Okay, you want to have a separate cutting board because obviously chicken contains a lot of salmonella bacteria and things like that. So you want to make sure that you keep this, at least use the other side of it or something. Okay, cut your vegetables first. Okay, whatever's not going to get cooked as much first, so that you don't contaminate it. But yeah, if you're not cleaning it regularly, okay, it could look like this, and not just on the surface. Remember, every time you cut something on a cutting board, what do you cut? You cut the cutting board. Okay, and so that means that bacteria and food items get pushed into the cuts, and that's harder to clean out. That's why every once in a while you should soak your cutting board in bleach. Okay, just for half hour or so, but soak it in a solution of water and bleach so that it kills anything that's even in those spots, because you can't clean those. Okay, your rag's not going to get in those cuts, things like that. Okay, you want to keep them. You got to bleach the whole thing, right? All right, does that sort of make sense to everybody? Okay, now we're talking here about bacteria that can hurt us. Okay. There's all kinds of bacteria, some that live in what we would consider to be the most inhospitable places in the world, yet they thrive there, right? It all depends on what kind of bacteria they are. We have found colonies of bacteria living in hydrothermal vents, okay, in the paint pots at Yellowstone National Park, okay, in the boiling mud, okay? We find them in there. Obviously, those kind wouldn't make us sick. They would never survive in our body. They'd die right away because it's too cold. Okay, or the conditions aren't right. It's not salty enough, or it's too salty, or whatever. Okay, every bacterium has a range of conditions in which it can survive. All right, so like we were saying here with the cell walls, bacteria cell walls are analogous. That means they serve the same purpose but not have the same structure as cell walls of plants, because cell walls of plants are made of, it's a type of fiber, cellulose fiber. Okay, whereas bacterial cell walls can be made up of a bunch of different things. The most common material is something called peptidoglycan. Okay, peptidoglycan is in bacteria that are known as gram-positive. Right? Gram-positive is it's one of the things. The gram test, sorry, is one of the things they do if they've given you a throat swab. Okay, they will run a gram stain on the bacteria that grow on the petri dish. If it comes out positive, you can have your infection treated with penicillin. If it comes out negative, then you would need like streptomycin, okay, or something like that, some stronger antibacterial antibiotic that would break down their different cell walls. Okay, so something that has peptidoglycan stains a certain color. Okay, it stains purple in the gram test, whereas a gram-negative cell wall typically stains like a reddish orange. Okay, those gram-negative cells cannot be killed with penicillin. Okay, all penicillin does is reacts with peptidoglycan and essentially dissolves it. Okay, removes their cell wall, and then they're easily killed off by your immune system. Okay, a gram-negative one, okay, that can be a problem, and it doesn't take much to produce a gram-negative cell. A simple mutation in a bacteria can produce a bacterium that is now resistant to penicillin. I think I was telling you guys this, but in like your university genetics, okay, you do one of your first labs is to irradiate E. coli bacteria. Okay, you put them under a UV light for 30 seconds. Okay, and under UV light for 30 seconds, you can mutate them enough that you will grow some colonies that are immune to penicillin. Okay, because all you do is you take those bacteria that you've grown over the last week, okay, you put them under the light, then you take them off of that petri dish and you swab them on a petri dish that has penicillin in it, and some will still grow. Okay, so it doesn't take much, okay, to kill them off, or to mutate them, sorry, so that you can't kill them off with penicillin. So yeah, it's a big deal. It's a health concern too, because obviously we have to develop more strong antibiotics, okay, things that can kill these off. So. All right. Okay, now, other places that we find bacteria. These are helpful bacteria, okay? They wouldn't necessarily make you sick. Soil, okay? Soil's full of bacteria, so you probably shouldn't eat it, okay? Don't eat dirt. I mean, it doesn't taste very good probably either, but okay. It's full of bacteria, and some of those bacteria have certain jobs related to like the nitrogen cycle, okay, other nutrient cycles and things like that that you might have learned about in like grade 6, okay? They help to change the form that nitrogen comes in. Okay, where's the most nitrogen present on Earth? In the air. Yeah. Okay, like 80% of our atmosphere is nitrogen. Okay, it's crazy, but yeah, okay, we don't even think about it. We think, oh, our atmosphere is made of oxygen, but oxygen is actually a very small percentage compared to the amount of nitrogen. So nitrogen is a very important gas, but the problem is plants can't use it directly. They need it. They can't make proteins without it, but they can't use the atmospheric nitrogen. So they have to have these bacteria, okay, that live in the soil convert it, okay, into other forms. They can also, there's also bacteria that help with the decomposition of, leaf litter, feces, other organisms, whatever, okay, that are on the ground, and they can help to release the nitrogen that's present in those, okay, in the nitrogen cycle. Right. So one of the forms of nitrogen that can be absorbed by plants is ammonium, okay, and the other form is a nitrate, okay, so NO3. Okay, so we have nitrogen fixing bacteria, which directly takes nitrogen and turns it into ammonium. Okay, and then we have nitrifying bacteria that do the nitrification part of it. Okay, so if you look at like compost, okay, how many people have the compost thing in their house now? Okay, have you noticed how much less garbage you throw out now? It's crazy. Like as soon as we started using it, like I feel like the black bucket now like half full, right? So compost obviously pretty good in that regard. It's also full of bugs and bacteria. You probably notice that in the summer. If you're not emptying it regularly, the fruit flies start to gather around it. Yeah, it's kind of gross. Yeah, my wife gets angry at me. There's fruit flies everywhere. Okay, so don't leave it too long, right? But there's all kinds of stuff in that compost that's helping to break down the organic material in there. Okay, and it's all important stuff. Okay, so you don't want to like spray it with something because you'll kill the bacteria. All right, so ammonium's an important source of nitrogen, but even more important is nitrates. Okay, that's what's in fertilizer, right? So if you fertilize your lawn and you see it starts to grow like crazy, okay, it's because there's lots of nitrates in that, so that's NO3. And there are bacteria that take the ammonium produced by one kind of bacteria and turn it into nitrates that's even better for the plants to use. Okay, so there's lots of bacteria around that are important for that. Okay, so in the nitrogen cycle, this is kind of the role that the bacteria will play. Okay, we have the nitrogen fixing bacteria that will take the nitrogen out of the atmosphere and turn it into ammonium. Okay, we have ammonifying bacteria that are taking humus, that would be leaf litter, feces, whatever, organic material, okay, and turning it into ammonium. And then we have the nitrifying bacteria that take that ammonium and turn it into nitrates so that plants can absorb it directly. Okay. Plants love the nitrates. Okay, that's what they can absorb. That's what they can use. Okay, so all of these bacteria have to be present in order for plants to grow. Okay, they live in the soil. Okay, they're tolerant to all kinds of conditions that the soil might throw at them. Okay, what do you suppose that picture's of? What's that? Okay, every one of these little yellow things is a Bacillus tetanus, in particular, bacteria. It's actually not a nail, it's the tip of a pin. The tip, not the head, the sharp end. Okay, that's the sharp end of a pin, okay, as seen under an electron microscope. All right, so when we look at it, we can only resolve it, to a certain resolution, right? It looks really pointy to us, and it is pointy, okay? But to an electron microscope, it's blunt, okay? Yeah. Everyone kind of follow me on that? It's just about how much we can zoom in, right? But look at how much bacteria is on the surface of that, okay? Probably all that happened, somebody handled it, right? They just, touched it with their fingers, right? What's the dirtiest thing in your house? Okay, I wasn't considering like just parts of the inanimate parts of the house. Okay, doorknobs are bad. Actually, you'd be surprised. Toilet seats tend to be clean bacteria-wise compared to things like keyboards, phone screens, and doorknobs. Okay, those things are absolutely disgusting. You know why? Because you look at the toilet and go, that's disgusting, and you clean it a lot more often than you clean your keyboard. Think about it. When was the last time you cleaned your keyboard? Okay, yesterday. How long before that? Okay, it's just we look at it and go, it's not a big deal. You're touching it with these all the time. These are gross. Okay, yeah, I'm gonna make you a germaphobe by the end of the day. Okay, so yeah, okay, anything that gets touched a lot, okay, is going to be really, really gross in terms of bacteria. Okay, I always give Mr. Dickey a hard time because he's a total germaphobe. Okay, like he'll open doors with like a paper towel in his hand. Like if he's leaving the bathroom, like as he's washed his hands, like he'll open the door with the paper towel and then chuck the paper towel. Okay, like yeah, he's really, really like intense about germs. He's got like hand sanitizer that would like I don't know, like take the skin off of your bones practically, and that he's always using. Yeah, but you got to watch out for stuff because stuff has bacteria on it. Okay, more so than you would think, and of course we can't see it. Okay, we don't know that. That's why you're always taught as a little kid: wash your hands, wash your hands, wash your hands. Well, you're always touching stuff. They're always covered in bacteria. Okay, like if you were going to use this pin to take out a sliver, right? You poke yourself with that. Now you got tetanus. But you guys get inoculated for that. But okay, that's the reason why. Okay, we get inoculated for tetanus. Tetanus is a very common bacteria that resides on lots of surfaces. If you get any kind of puncture wound, okay, you're likely to get a tetanus infection, right? And well, tetanus is not a good way to go. Lockjaw, exactly. Tetanus, the bacteria releases a toxin that does not allow your muscles to relax. So you clench your muscles and they won't unclench, and they just get tighter and tighter and tighter. You can break your own jaw. Okay, you can't breathe because the muscles that enable your breathing just contract, and then you can't breathe. Okay, it's not a nice way to go. So we have a vaccination for it. So you should get vaccinated for that. Vaccinations are good. Okay, and if you're going to take out a splinter with a pin, what should you do with it first? Okay, dip it in bleach, wash it, okay, but then rinse it after the bleach, okay? Or you just as simple as like just if you have a lighter, just like run the lighter under it a couple of times. Doesn't it? You don't have to get it like smoking hot, okay? It doesn't have to glow or anything, okay? Just run the flame over it a couple of times, okay? It's good, right? Yeah, just take your finger and wipe it off. That won't work, okay? Don't go. Yeah, that's not going to do it either. That's just made it worse because you just breathed all over it. Okay. So yeah, you got to think of things like that, right? It's why if you go to the hospital, they don't just reach in the bucket over there. It's like, oh well, I'll just use that needle again, right? Like that's why we don't do that. Okay. You wouldn't believe how easily contaminated things are. All right. It's why that when the doctor comes in, if you've ever had surgery or anything like that, he has someone put his gloves on. Right, he gets sterile. Okay, so he washes really thoroughly with really hard soap. Okay, and then he has someone else who's also done that, put his gloves on for him. Okay, things like that, so that they're not contaminating anything. Right, they take a lot of precautions with infection because bacteria can survive anywhere, and they can survive there for a long time. And even worse than that, they can go dormant. They can go dormant for hundreds of years even, okay? And then as soon as the conditions are right, they'll grow again, okay? So we gotta always be careful of things like that. But they can survive in lots of habitats that no eukaryote could possibly tolerate. Okay, so prokaryotes do not have membrane-enclosed organelles. All right, so if we were to look inside, this is an electron microscope picture of what a bacteria, like a tetanus or a salmonella bacteria would look like. Okay, and what we can see, they have ribosomes. Okay, they have to make proteins, so they do have those, but those aren't really membrane-enclosed organelles. Okay, they have a capsule, which is kind of like their cell wall, okay, or part of their cell wall. They have a plasma membrane, okay, so they do have like a cell membrane. Okay, they have a nucleoid region, so their DNA is just kind of there. It's not really protected by anything. Okay, and they have mesosomes, which help them with cell division a little bit, but do you see lysosomes or Golgi apparatus or anything like that? It's just not there. All those processes that those individual organelles do in their individual compartments all have to be done in the same place, which means it's not very efficient. Okay? If you have to do all of those jobs in the same place, it's not going to be done well, which is why you don't see prokaryotes getting as big as eukaryotes or as complex as eukaryotes. Okay? Without the added protection of the nuclear membrane, the DNA is very easily damaged in a prokaryote, okay? Which can lead to its death or its mutation. Okay? Prokaryotes and eukaryotes are both capable of repairing damage to DNA. The problem is they don't always repair it right. Okay? And now there's some change in the DNA, and when you're something small like this, okay, that can lead to fairly significant changes in the way the thing functions. All right. They also have what are called pili. Okay, there's these little kind of tentacles that they can use to grab onto things. Okay, attached to the inside of attached to a cell membrane of another organism, or the inside of a blood vessel or something like that. Okay, and then they have multiple flagella. Okay, that they use to move around. Okay, and we've seen this diagram before. Prokaryotic cells only have one chromosome. Okay. You and I have 46, okay? But a prokaryotic organism will have one chromosome and it's shaped like a ring, okay? It's shaped like a ring when they're dividing. When it's not dividing, it's just like ours. It's snarled everywhere, okay? So when they divide, they have to copy that DNA and then it gets to both daughter cells, okay? The big thing here with prokaryotes is that in times of stress, they can go dormant. They form what's called an endospore. They essentially just go into a state of complete suspended animation. They have no metabolism whatsoever. Okay, they're just completely dormant. And then if they were inhaled in dust, okay, or something like that, then suddenly their environment changes, they trigger, and they come back, and they can cause severe infections. Okay, it is believed that this is what happened with the curse of Tutankhamun. Okay, you guys know who Tutankhamun was? King Tut? Yeah, the Egyptian king. Right, one of the first pharaohs whom they found his tomb intact. It hadn't been raided. They found his sarcophagus, okay, with the body inside. So the Egyptians, when it was discovered, told the archaeologists, like, don't go in there. Okay, like, Tutankhamun has a curse on him, and as do all the pharaohs. They're not supposed to be disturbed. If you disturb them, really bad things are going to happen. Of course, they didn't listen, so they went in and they opened the sarcophagus. Now, the problem with doing that is that there would be a massive difference in the air pressure inside and outside, just due to differences in temperature. So when they opened it, there was an overpressure inside, and dust from inside blew out into the faces of the archaeologists. Okay. Within days, anyone who was there when the coffin was opened was dead. They all got sick and died. And the Egyptians went, "Told you so. You shouldn't have opened that." Okay? But what they found later was that these people died of a rapidly reproducing bacterial infection. Okay? And it's believed that there was someone who, possibly prepared the pharaoh when they were doing all the mummification stuff, whatever, may have had that bacteria as well. It was stored in there. When they opened the sarcophagus, the dust contained the endospores. They inhaled the endospores, and the bacteria grew inside of them. Okay, so they can go dormant for long, long periods of time. Okay, prokaryotes for the most part are chemoheterotrophs, but there are also some that are photosynthetic. Okay, those are generally like blue-green algae, very simple types of algae. Okay, so chemoheterotrophs means obviously that they have to consume organic molecules for food, which is why they like the inside of your body so much. There's lots and lots of raw materials available in there for them to feed on. Okay, now what is it about a bacteria that makes you sick? Actually, I mean, it's a little bit your body trying to fight it, but it's not like a virus. Okay, like a virus makes you sick by making your body kill its own cells. Okay, in an immune response. And a bacteria, they're bigger, so your body goes out and actively hunts them down without killing your own cells to do it. Okay, what makes you sick from a bacteria is their waste products. Bacteria waste, generally their waste products that they produce are toxic. Okay, they release poisons into your body that make you sick, which is why bacterial infections often have different symptoms than do viral infections. Okay, viral infections, typically kind of go through the same kind of gamut, but a bacterial infection has usually different symptoms, and a lot of times they're more intense because of the chemical nature of your sickness versus the microbiological nature of it. Okay, so you've got these things that are releasing toxins, especially like salmonella. Anyone ever got food poisoning? Salmonella? Like, it's horrible. Like, and it can kill you if you get a really serious bout of it, or your immune system is depleted. You can die from it. But it's horrible. Like, you're like 2-3 days where you're in the bathroom for 2 or 3 days, and you don't know whether you need to sit on the toilet or face it. Okay, like that's how awful it is, right? But it's because the chemical that salmonella bacteria release irritates your digestive tract. Okay, makes you nauseous. Okay, makes you want to throw up. Okay, unfortunately, it's not like having the stomach flu, which is viral. Okay, where after you throw up a little bit, you feel a little bit better. Okay, you just keep throwing up, and you just feel awful. Like, yeah. Don't get food poisoning. It's awful. Nasty. Okay, but they are chemoheterotrophs, and they're just taking your stuff. Yeah, yeah. I mean, unless you want to live dangerously, up to you. Okay, now difference here, guys, is obviously eukaryotic cells have a true nucleus. There's a special membrane that surrounds the genetic material, keeps it safe, protects it. Okay, and they have other membrane-enclosed organelles, and that's advantageous because many of the functions of the cell, especially digestion, require different environments to function effectively. All right, if we've got no place, or if you're looking at a prokaryotic cell, it's got to digest its food in the same place it keeps its DNA. Okay, and that's not good. Okay, I mean, you got these harsh chemicals that you use to digest food, and you got that in the same place where your DNA is kept. It could damage your DNA, or the process isn't going to happen very quickly because you can't have those chemicals very concentrated. Right? You've got to burn sugar in the same place where you're processing wastes. Okay, you don't want to be doing those two processes together. Okay, that's like going to the bathroom in the kitchen. Okay, you keep those things separate. Okay, and you keep them separate for a reason. Okay, they're processes that aren't compatible with each other. right? So that's why a eukaryotic cell is generally larger, more efficient, okay, than a prokaryotic cell. It does have separate environments for all of those processes. Okay. Also, specialization is a big thing. You've got to have organelles to become specialized. Okay, although eukaryotes are less diverse than prokaryotes, there's way more species of prokaryotic organism than there is eukaryotic organisms. Okay, the complexity of a eukaryotic organism is vastly superior, okay, to that of any prokaryote. Okay. All right, so how did they come to be? There's several ideas about how that happened. Okay, the first theory is that is the invagination theory, and that means that the cell membrane just folded in on itself and created separate pockets within the cell that eventually became the organelles. Okay, there's a fair amount of like fossil evidence to support that. And the other one is the endosymbiosis theory. Okay, and that is that several prokaryotes actually merged together to become a single celled eukaryotic organism, right? So you might have had, a ancestral prokaryotic organism that was really good at, breaking down sugar, carrying out cellular respiration, and another one that was photosynthetic, okay, and some sort of, larger kind of host cell, okay, they came in there and they started working together. There's also evidence to support that theory, okay? A lot of the evidence there is that there is actually genetic material in some of your organelles in the cell, okay, especially in the mitochondria. Okay, there's actually mitochondrial DNA in the mitochondria that is different from the DNA in the nucleus, okay, which would seem to support that perhaps mitochondria were their own organism in the very, very distant past. Okay, most scientists believe it's some combination of both. Okay, that a little bit of both happened, okay, to produce true eukaryotic organisms with organelles. Okay. No one's probably ever really going to know, okay, but it's probably some combination of both of those things. All right, I'd like you guys to give me some answers to those questions. You got some space there, okay, so you can answer those questions, and then we'll go over them, and then we'll talk about something else here. Okay, so we want to look at, first off, why are most prokaryotic cells so small? They're not what? Yeah, well, they're not complex, and they don't have compartmentalization. Without compartmentalization, they can't perform the basic functions of the organism as efficiently as a eukaryote could. Okay, second question. What might be the reason why there are no multicellular prokaryotic organisms? It would require them to become specialized, and you can't become specialized if you're not compartmentalized. Okay, it would be impossible. And for question number 3, why can prokaryotes survive in hostile environments that would kill a eukaryote? Okay, partially they can go dormant, but there are some that would actually like thrive. They would live. Yeah, they can adapt quickly, but also because they don't have this they don't have the compartmentalization, they don't have to maintain the same kind of environmental conditions within the cell that a eukaryote does. Okay, because of that, they can actually perform, even though it's inefficient, they can still perform those basic cell functions even in environments that would not be hospitable to a eukaryote. Okay, it would be impossible for a eukaryotic cell to maintain all the separate environments, let's say in a really salty environment where osmosis would have water flying out of the cell. Okay, a prokaryote, with that peptidoglycan cell wall, is not as susceptible to those conditions. All right, everybody all right with that? Okay.